Hi, everyone. My name is Lu Chow. My pronouns are she, her, and hers, and I'm here today to talk to you about some of the work that we've been doing at NASA's Goddard Space Flight Center. I'd like to thank Dr. Chris House for inviting me today. It's been a real pleasure to meet all of you and to speak with all of you. I usually like to, well, one thing I'd like to premise with is that it's going to be a little bit of an interactive talk in the beginning. And I'll go into more details later. And so if I can ask for your participation as you think about some of these questions and the answers that you might have for yourself. And for anyone who is online, if you have questions or answers in the chat, I think, Dave, you're on it. Okay, awesome. Okay, so I'd like to start off my talk with this image here to kind of hone you all in on this cosmic perspective that we have. So I'm an astrobiologist. I like to think about life and the life that we have in the universe. And as we look at this image, you kind of want to, remind yourself that all of the life forms that, you know of, so that's you and your relatives and the microbes that live on you and your relatives, the microbes that live in, these extreme environments that you hear about in Antarctica or on volcanoes or in really dry deserts, or those really exotic animals that you see on the latest BBC documentary, in the deep sea oceans, near hydrothermal vents. Those are all on this one single image here on our planet. And as you focus on your mind on this perspective, this cosmic perspective that we have, I also want you to think about how we as humanity observe those life forms. Do we see them with our own eyes? Do we use techniques to look for them? Or how do we recognize it as life? And I like to kind of draw on this image here to the left is, does anybody know what the left one is? You can shout it out. Tardigrade, a water bear, right? They're these tiny little organisms that are super, super cool and super interesting. They can survive all the crazy stuff. But we can't really see it with our own eyes. We need really sophisticated instruments like a scanning electron microscope to take an image like this. In contrast, on the right here is an image of the deep space. So this is Webb's first deep field. So every single dot in that image here is a galaxy that was taken recently on the James Webb Space Telescope. So as you peer out into into this image on the right here, you kind of have to grapple with this micro and macro different versions of the universe and the kinds of techniques that we use to observe them. And so as you think about, finding life forms like tardigrades, for example, or looking at one of those tiny little dots and asking the question of, is there life on that one of those dots? I'd like you to think about the characteristics of life. How would you identify life if you were the astrobiologist developing a NASA mission, for example? So that's the question. I'll leave you to a few minutes to think about what are some of the characteristics of life that you can think of, and raise your hand if you have any thoughts. Yeah. It has DNA. Yep, that's a good one. Yeah. It can reproduce. Yep. Remember from high school biology. Anything else? Yeah. It's carbon-based. Yep. Yep, it does. It does. It needs to consume some sort of matter to survive. So it eats things and it, produce wastes. Yep. Any other characteristics of life? Yep. So those are all really great answers to this question. And as you can already kind of see that there isn't really one thing, right, that you all jump to that's like, oh yeah, this is life. Like you don't just raise your hand and be like, oh, everybody knows that this is life. There, these different characteristics of life are part of how we try to answer this question of when we go out there to look for life on other planets in the universe, what can we actually focus on? And typically you want to kind of define it first. right? When you want to look for something, you kind of need to know what it is. But we actually don't know what it is. We're pretty advanced in terms of our intelligence or civilization, but we don't have an accepted definition of life that's agreed upon by all academics in the world. And so there's a great quote that I like to bring up by Dave Desmarais, who's at NASA Ames. He said, if you have 100 people in the room, you're going to have 150 different definitions of life. And this is absolutely true, because people can't really seem to agree on what that definition is. But I'd like to challenge you into thinking that we don't necessarily need that definition of life. We could potentially use an operational definition of life, something that can give us a starting point when we go out there and design missions to look for them. And so instead of asking you what are some of the characteristics of life, like whether it's it has DNA or it reproduces, what are some of the imprints of life that you can think of? Imprints of life that is made and can be observed in the environment. And some of those overlap, right? And so there's no wrong answers. So just imagine what you think are those differences. Take a few minutes to think about it, and then I'll raise your hand. Yeah. Yep, exactly. So fossils. That's one imprint of life. So if you go out there to another planet and you see a fossil, you might want to question how that fossil got there. So that's one physical like manifestation of life. Anything else? Yep, exactly. Yep. So the oxygen we can detect in our atmosphere, right? So all of the O2 we have in our atmosphere actually came from life. Well, I shouldn't say all. Yeah, pretty much. So yeah, tangible things that we can actually observe or that we can design instruments to observe. So one of the answers from before, the characteristics of life, is reproduction, right? Reproduction is a process, it's a characteristics of life, but how do we actually observe that reproduction? And so maybe observing, I don't know, cells that are dividing or organisms that make more of itself, that would be an imprint of life that we can observe and see, but reproduction is the process that is intrinsic to life itself. I forget where my next slide is, so let's just go and see. So this is the outline of my talk. I'll be discussing how we search for life, especially life as we don't know it. And so all of the characteristics that you've mentioned and all the imprints that you've mentioned came from our understanding of life on Earth here. But I want us to remove that bias a little bit, because there's no formal theory to say that life has to look like what life is here, even biochemically. And so I'll talk about what that means and how we search for it, and how we define it. And so Terran biosignatures are biosignatures that are based on Earth. So we make Terran biosignatures. Agnostic biosignatures, which is the team that Dr. House and I and the folks at Georgetown and NASA are a part of. We'll go over what that means. And then I'll also discuss some of the instrumentation that we've built to look for life on other planets, and also the future missions that will have those instruments on there. So some familiar examples are some of the things that you've already mentioned: DNA, RNA, specific compounds, so metabolites, things that you ate for breakfast. That's, what you ate. And so you metabolize it, and that's, a biosignature. Homochirality is this idea that life is one-handed. So all of our amino acids and sugar that biology has, for some reason, likes to be in one kind of configuration. Chirality is just compounds that are mirror images of each other. It's a really intriguing problem in astrobiology. We still don't know why life chooses one over the other. Isotopes are just two different compounds that have the same protons but different numbers of neutrons. So one is a little heavier than the other. For some reason, again, life likes to use the lighter one sometimes and maybe the heavier one sometimes. And so we can look at the ratios to kind of determine whether there's been a biological process that happened. So those are all chemical biosignatures, minerals and gases, as someone already mentioned. And physical biosignatures are the ones that you can observe in the physical realm. So things like fossils, dynamic movement. So things that are moving in a certain pattern that is different from just random like wiggling around. And even spectral signatures. So colors could be a biosignature as well. Some of these examples I already mentioned: homochirality, life likes to use one type of amino acid. Isotopes, for example, life is known to fractionate a certain type of certain isotopes to produce distinct biosignatures. But the problem is some of these have also been found to be made in abiotic environments, so environments that doesn't have life in it. So in space, meteorites, we found an excess of one-handed configuration of amino acids over the other. So that's super interesting, right? So we can't really just go out there and say, oh, there's one type of handedness over the other, therefore we found life. We know that it can also be produced by abiotic process, and same with isotopes. DNA, RNA, and proteins are biopolymers. So these are just long chain stuff that make us up. They're used for information storage and for functionalities. So that's how we work. We're just a glob of proteins glommed together, doing things all the time. And these are really good biosignatures because the likelihood that these polymers form in long strands are super, super low. And they're not likely to be formed abiotically. And so if you do go out there and you do find long strands of DNA, you probably have found life. But actually, you probably have to first ask if you brought it with you before you can say you found life. Some physical biosignatures examples, stromatolites. These are really cool rocks, microbially induced sedimentary structures. So when you have life and rocks living, coexisting in an environment, the life can do things to it that are really weird and wonky. And this is another one of the fun little exercises I'd like you to kind of question. So do you think that the one on the left is made by life, or the one on the right is made by life, or both, or neither? You don't have to know the answer. Just guess too. There could be someone in this room who knows about stromatolites. I'm guessing someone over there. Yeah. That is life or not life? No, it's the one on the left that is life. So stromatolites actually grow upwards. So we have these like cyanobacteria. It's okay. It's okay. It's really difficult. It's really hard to do life detection, right? Especially when you encounter these things. And so if you're a robotic explorer on Mars, for example, and you see them, you have to do a lot of interrogation to kind of get to that answer at the end, whether it is life or not. And so, and it's not just by looking at it either, right? You have to do chemical analyses. You have to do all these different types of analyses that can inform you on how likely is this pattern to be formed without the presence of biology versus this one. And so this is the stromatolite. It likes to grow up towards the sunlight because they're made out of mostly cyanobacteria a lot of times. That's ingrained with silica particles and all those things. And then the right is a metamorphic rock. Just a rock. And then lastly, another familiar example that I like to bring up and we can't forget about is signatures that we can detect remotely. So anybody has seen the movie Contact? Yeah. Carl Sagan's Contact, yes. Good movie, very good movie. So remote biosignatures are biosignatures that we can observe from Earth of somewhere far away. So that can be a different planet in our solar system or an exoplanet, a planet outside of our solar system. Someone already mentioned before, gases in our atmosphere could serve as a biosignature. And so oxygen and ozone in our atmosphere is one of the great biosignatures that we can observe if there's life elsewhere that also makes oxygen. What are the chances? And methane as well. There's this really cool thing called the vegetation red edge, which is this idea that the vegetation cover of the Earth absorbs everything but the green light. And so there's an edge when you look at the spectra from a spacecraft, for example, and that edge is very, very distinct and can tell you that there's vegetation cover. made out of organisms that can make chlorophyll. So that's cool. And of course, we are biosignatures. The narrowband pulse radio transmission came from us, the only civilization that I know of that invented the radios. So that's really cool. We can develop these techniques to look for these different biosignatures. But the most important thing that I want you to take out of all of this is all these examples that I describe has this really strict contingency to the history of life on Earth. And so the fact that we evolved as an organism and eventually, photosynthesis came about. So you have the vegetation red edge, right? So that's based on the history of life on Earth and the history of our planet. The same with humanity in general too. Like if we make these, pulse radio transmission, we had had to have survived all these, from our ancestors. The dinosaurs had to have died, and we had to come to be, and then we'd have to grow a big enough brain to develop radios. All of that is contingent on the history of our planet, the circumstances that we have here on our specific solar system. And so it begs the question of, well, then are all these biosignatures specific to just this planet? And so I'd like to walk you through this kind of like thought experiment a little bit, that if you have, this is a geologic history, geologic timeline. Yeah, geologic timeline, starting with now. So the Holocene, spiraling all the way down to the beginning of, what does that say, 4.5 billion years ago, which is when the Earth was formed. And so if you were an alien civilization and you came to our solar system, and you happen to be here in 2023, and you look, you observe all this vegetation red edge and ozone and all that stuff, and even radio pulse transmission, you only see those things when you're here in the Holocene, right? So what happens if you ended up like back here 65 million years ago, when the dinosaurs were still around? Would you see dinosaur biosignatures? Probably. What about back here in the Precambrian, when life was a lot more simple? What kinds of biosignatures would you see? You wouldn't see all of the things that I just described to you, right? You might see DNA and RNA because that's deep rooted within our biology, but you might not see those things that I mentioned, radio signals and things like that. And what about even further before, in the Archean, when life was super simple? And so let's talk about agnostic biosignatures. How do we remove ourselves from this bias that we have for life on our planet? And so I want to start off with this, walking you through this this concept of biosignatures. So this gray oval here is, I am calling Terran biosignatures. So biosignatures that are on Earth that we know of and that we are familiar with. Cool, great. A subset of that is probably going to be shared with other life forms, because other life forms are life, if there is any out there. And so conceptually, there's some features that we have that's going to be the same as those features of those life forms. And so that's what I call universal biosignatures. We have no idea what universal biosignatures are, because again, we haven't found life elsewhere, so we don't know what those common features are. But there must be common features because we're life, and then there's non-life. And then so here in this gray bit is what that history that I talked about, the historical contingency that depends on our planet. If you go to another planet, you might have a different historical contingency. If you go to Mars, you have a totally different history, totally different dynamics, whether it's geology or atmosphere, hydrology, or astronomy even, where it is in the solar system. That can drive different evolution of life if it started differently or even started the same. And so each of these petals in my little flower is its own planetary bodies, and the history of life that are on those different places. Agnostic biosignatures attempts to move from that bias of being further away from the center of the flower towards the center of the flower. There's no right answer to what an agnostic biosignature is. I see it as like an onion shell, where being really, really specifically focused on the outside is our Terran bias, thinking that life must look this way. And as you take off those shells, you get closer and closer and closer to what could potentially be commonalities between life here and life elsewhere. And that's the characteristic that you want to focus on. So again, just going back to these chemical biosignatures and physical biosignatures that I talked about before, what could those look like in an exotic system as we get closer and closer to as we get more and more agnostic? So I want to do a thought experiment. Okay, nice thing, right? Here's an image of Mars, I think. Yep, I think it is. And let's say we find a paperclip on Mars. So imagine that you don't have the knowledge that you have now of what a paperclip is or what it looks like even. How would you know that this paperclip was made by life? As opposed to like a rock right here next to it. How come this is made by life and these are not made by life? So take a few minutes to think about what are the characteristics of this paperclip that makes it different enough from all these other things that you could invoke the presence of something interesting or a process to make it. Yep, we recognize the shape. Yep, that's one reason. But if you didn't, like if you don't know that that's a paperclip, right? Yep. So the likelihood that you'll find a concentration of a certain type of, alloy or steel or aluminum or whatever, is a lot lower than finding just generic rocks that we know are easily made in these types of environments. And so I like to think of this thought experiment in terms of steps. How many steps does it take to make this rock versus how many steps does it take to make this paperclip? So with the rock, you kind of need to have the right elements. You kind of need to have the right temperature, the right pressure. They have to be exposed on the surface. And different rocks, different planets, of course. Got to remember that. So you kind of have to also know the baseline, right? There could be a rock that's super exotic and totally shouldn't be there. And maybe you should be asking whether or not that rock was made by life, like the stromatolites, for example. In the paperclip, on the other hand, you need all of those things, right? The right elements, the right pressure, temperature, surface, be exposed to the surface. You have to mine it for whatever metal is made out of. It has to be purified. It has to be molded into thin shapes, long, skinny things, bent and folded. And probably most importantly is you need the instruction on how to make a paperclip. So that information has to have already existed there. And that information I find analogous to life to turn to take the step from 1 all the way to 9 to make a paperclip. And so the likelihood that there's a rock that you see on Mars is high, and the likelihood that you find a rock or find a paperclip on Mars is low. The unlikelihood of that suggests that there could be another process that's not just geochemistry or geophysics or I don't want to say random because it's not random. Like physics is physics; it's still driving things. But something that is invoking a process that contains information that can be passed on to the future paperclip makers. And so you can also apply the same analogies to molecules, right? And so this is one of our favorite organic geochemistry molecules, the styrene. That's a styrene, right? Yes. And you can think of the same way, the steps of making this molecule. So instead of looking for specific molecule like this one here, we look for how many steps it takes to make them, how complex they are. The likelihood that you find something like this in space is maybe a lot higher than finding something like this in space. And so conceptualizing this whole idea of how many steps it takes to form the molecules is one approach to agnostic biosignatures. So we don't need to know if this is made out from biology. This styrene molecule requires upwards of 40 enzymes, right, to catalyze each of these different steps to make it. And so invoking the need for those enzymes can kind of tell you that this requires a lot more than just the environment. So when you go to places like Europa, for example, or even the cool hydrocarbon seas of Titan, we need to start thinking about how we can extrapolate the search strategies that we already have for Terran biology or Terran biosignatures to agnostic biosignatures. And to do this, we use an instrument called mass spectrometers. And so I'll walk you through the history of mass spectrometers first, as they are used in space. And then we'll talk a little bit more about how we can use them to look for agnostic biosignatures. Mass spectrometers, what are they? Has anyone heard of them before? More or less? Yes. Yep. They're kind of they're in almost every chemistry labs you can imagine. They're the bread and butter of how we can characterize organic molecules or other kinds of molecules or particles or inorganic stuff or gases and whatnots. And the way that mass spectrometers work, there are just 3 different parts of it. That's all you got to know. There's an ionization source, which is just a device that turns a molecule into ions. It puts a charge on it because we can manipulate the charge, but it's harder to manipulate the ions. There's a mass analyzer, so it filters out those ions, so molecules that have been ionized or broken up into smaller ion pieces. It filters them out by mass. So it tells you that, oh, this is has a mass of 12, which is a carbon. This is a mass of 15, which is a nitrogen. And then a third part of it is a detector. So that's just counting the number of ions. So I have 100 of carbon 12 and then 1,000 of nitrogen 15. So mass spectrometers are these incredible tools that we've used previously to characterize atmospheres of planets and surfaces of planets, or send them to the ISS. That's where we also have mass spectrometers. Missions like the Pioneer Venus were the ones who pioneered the use of mass spectrometers to characterize planetary atmospheres. Vega, Galileo, which was used to look at Jupiter, and Stardust looked at a comet. Maven looked at Mars, Juno, Jupiter. These future missions, JUICE, Europa Clipper, and Da Vinci. JUICE for Jupiter, Europa Clipper for Europa, obviously, which is the moon of Saturn. No, a moon of Jupiter. Sorry. And Da Vinci is a mission to go to Venus. All of those different missions houses a mass spectrometer on board. We also have ones that have looked at both the planetary atmospheres and the surface. So Venera, Cassini, Huygens, Rosetta, Philae. Cassini-Huygens is one of the bigger missions out of NASA that went to Saturn. It characterized Saturn's atmosphere as well as Titan's atmosphere and Enceladus. Huygens is a probe that was sent into the towards the surface of Titan, the first probe that we ever sent to Titan. We're going back in a few years by the Dragonfly mission, which is an octocopter that also houses a mass spectrometer. The Mars Science Laboratory, which is the mission that I'm a part of, has been roving around Mars for the last 10 years. It's been really incredible and exciting, and Dr. House is a pretty big part of it. He just made some really intriguing discoveries of the methane that we've observed on the MSL mission. Back in the 70s, this seminal mission, the Viking missions, was the first lander to go to another planet that has mass spectrometers on it. And it contained these 3 biological experiments that was our first time really ever to try to look for life. It's the first life detection mission. We haven't done really anything close to that since, but we're hopefully going to in the future. So yep, these are what I already mentioned. It has a gas chromatograph mass spectrometer that's part of this biological experiment. The Phoenix lander has an evolved gas component that can look at the gases in the Martian atmosphere. It also looked at the types of materials that's in the soil. And so we kind of learn a lot about the salt and the oxidants that are in the soil that told us how to make our future missions better. Venera measured the chemical composition of Venus, and the Venus bus probe is one of the American ones that measured the composition of the lower atmosphere of Venus. All of these using different types of mass spectrometers. There's different flavors too of mass spectrometers, and so you can see the little symbol of what they are. A quadrupole mass spectrometer just uses an AC/DC charge. I probably shouldn't go into the details, but it's just used like 4 rods to drive the electronics. Magnetic sector uses magnets to separate the masses. Curiosity, Mars Science Laboratory, has found some really, really fascinating stuff on the surface of Mars, including these chlorinated organic compounds that can tell us that there's carbon indigenous to Mars and chlorine indigenous to Mars, in addition to sulfur-bearing materials that could tell us a lot about the preservation of some of those organic matter. Whether or not it has life, we don't know. So far, there hasn't been any indication that these are biosignatures. These are a lot more simple than our biology likes to have. Oops. And I already mentioned the Cassini-Huygens probe measured the upper and the lower atmosphere of Titan, and we found these really interesting. I mean, I shouldn't say found. We've thought about what kinds of organics that could be present on other planets a lot, and Titan is one of those places that we kind of call a planetary laboratory. Organics have been found to be made in the atmosphere and coalesce to form these bigger and bigger blobby stuff. And these are called tholins. And these tholins are organic rich, right? And by organic, I mean carbon-based. Whether or not those carbon-based material can form life or is the precursor to life, we still don't know. Hopefully we'll find out a lot more when we go there in the future. So you know now what mass spectrometers can do, and you know that it's been a huge part, this huge heritage for a lot of the missions that we've done in the past. So how do we go back to this whole idea of agnostic biosignatures, and how do we infuse those two approaches together? How do we use something that we already have, a tool that's already been built and sent to space and over and over and over again to look for agnostic biosignatures? So one way to do this is, again, going back to this idea that we can think of those biosignatures as steps, or how to form these molecules as steps. So one of the examples that I want to point out here is that, so the left molecule here is a fatty acid, and the right here is a styrene that I kind of already alluded to before. Fatty acids are putative biosignatures, which just means that they're kind of biosignatures sometimes, but not all the time. So there's ones that can be made abiotically, and then there are ones that are much harder to be made abiotically. And so we think that it's could potentially point to the presence of life. Whereas stearenes, on the other hand, it's just impossible to make something like this without the presence of biology. Oh, 30 enzymes, that was close. And it requires molecular oxygen in the atmosphere to make. And so our contemporary interpretation is that maybe this is a biosignature, and this is definitely a biosignature. But we needed to have known that those are fatty acids, right? We needed to have known that those are stearenes that are made from these 30 enzymes that I mentioned before. We needed to have the knowledge of the biosynthetic pathway to form them. An alternative interpretation, again, is this idea that if you can look at the complexity, the number of steps that it needs to make a molecule like that, you can see that fatty acids have a lower complexity than something that has a higher complexity. And so this work is being developed, or has been developed, by our collaborators at University of Glasgow in this latest paper on how to identify these molecules and using mass spectrometry, right? So mass specs are used to analyze compounds like this. And so they came up with this idea that when you have a compound like this and it breaks up into smaller pieces, the number of fragments should reflect the number of steps that it takes to make them. Now, a lot more work needs to be done to address whether or not this application can work with the types of low fidelity mass spectrometers that we send to space. By low fidelity, I mean we can't just have like our big, beautiful mass spectrometers that we see in our benchtop versions, right? We have to miniaturize them. We have to develop ways to work on them when we're millions and millions of miles away. And so those mass spectrometers are great at answering the questions that we have about what kinds of chemistry we're encountering, but they're not as great as if we had the sample with us, right? And so that's why return missions like OSIRIS-REx can also be a really critical tool in life detection in the future. There's also another way to look at agnostic biosignatures in terms of patterns. And so this idea called the Lego principle was dubbed by a relatively well-known-ish astrobiologist in our field, Chris McKay. And he said that life is made out of building blocks, right? And so when you go out there and you find these building blocks, and you find those building blocks in really high abundance, then you can kind of guess that there's a process making those things in high abundance. And you need, again, the presence of information in the first place. But it doesn't have to be that same building blocks. It doesn't have to be amino acids. It doesn't have to be nucleic acids. What if it's something else? And so what if you go out there and you see the distributions of molecules that look random or uniform? You can kind of tell that perhaps there's no specific process that's driving it. But if you see these distinct patterns, these like peaks that are spiky, maybe something interesting is going on. And they don't have to be those molecules that we know and love. It could be anything. So this is a example of that patterns of biosignatures that I mentioned. So amino acids is one good example. We happen to pick 20. We are made out of 20 canonical amino acids, 2 non-canonical, I guess. But there's hundreds more of different types of amino acids that's been found in meteorites, for example. And so why the 20, right? And so if you go out there and you say, oh, I didn't see the 20, so there's no life. Maybe that's not the approach that we should take. Maybe we need to look at the abundances of those amino acids. And same with lipids. And so lipids that are made by us are made from a specific biological pathway that adds on a specific number of carbons. And so there's 2 different pathways, one that's 2 and one that's 5. So if you go out there and you see fatty acids that are, for some reason, has these even over odd ratio where there's more of the doubles that are different by 2 instead of by 1, then you can kind of guess that maybe something interesting is going on. But again, it doesn't have to be this specific biological pattern, right? It could be something else that could be driving the overabundance of one type of organics over the other. And so this is an example of the agnostic biosignature patterns that I was talking about. To the left here is a mass spectra of the Murchison meteorite extract using a type of really, really fancy mass spectrometer that we haven't miniaturized to send to space yet. And you can see already that the distribution of the compounds that are in there based on their mass shows that it's uniform, right? And so maybe that uniform distribution can tell us that there's a difference when you look at things like something like an E. coli extract, where you see these like spiky patterns of the concentration of certain types of molecules that are in there. And when you rank them in an order, perhaps there's a distinguishing of a pattern. And so if I had erased this Murchison extract and erased this E. coli extract, I don't know what those are made out of. Can I just look at these 2 lines to tell what whether one is life or not? And so that's one way to think about agnostic biosignatures using mass spectrometer. And this is data that I collected using a mass spectrometer called the Mars Organic Molecular Analyzer that will go on the ExoMars mission eventually. All right, and lastly, let's talk about some of those really cool missions that will happen in the future that hopefully some of you will be a part of. So I already mentioned the ExoMars rover. This is the MoMA instrument suite. It's a laser desorption ionization mass spectrometer that's capable of trapping ions and performing tandem mass spectrometer experiments. So that's just fragmenting your ions into smaller pieces to then put it back together to figure out what you're looking at, and potentially even use that agnostic biosignature that I mentioned. How many steps does it take, or how many fragments does it have to see whether or not it's something that is complex enough that it requires life. So that's the next generation ion trap I already mentioned, the laser source, the laser desorption source, the mass analyzer, and then the detector, and then you get this like nice mass spectra at the end that can tell you what it could potentially be. The Dragonfly mission, this one's a really cool one. It's an octocopter, octo meaning 8. There's 8 thingies I don't know what they're called. So this probe is going to go to Titan by itself. There's not going to be an accompanying orbiter or anything. And it's going to land on the surface of Titan. It's going to land in the equatorial region around the dunes. And it's going to characterize the surface of Titan to see if there's habitable environments that is conducive for life. Maybe life as we don't know it, because it is dang cold on Titan. It's 94 degrees, which is minus 200 something Celsius, degree Kelvin, so minus 200 something Celsius. And so you can kind of imagine that if there is life forms out there, it could be totally different. And there's no hydrology, as in water cycle there. It's actually the lakes and seas of Titan is made out of liquid methane and ethane. And so you can imagine already that that's a radically different chemistry than ours, that if there is life on there, you would need some sort of agnostic biosignature tool to detect them. Da Vinci, the deep atmosphere, Venus, investigation of noble gases, chemistry and imaging. It is an atmospheric probe. I was selected last year or maybe 2 years ago now, as a mission to go back to Venus to look at the atmospheres and determine if there's anything we can glean from the types of gases that are in Venus's atmosphere. Noble gases is an important part of trying to understand the history of the planet. And because I'm an astrobiologist, I also like to think about cool gases that could be made by life, whether it's life on Earth or life elsewhere. And so recently there's this findings or discovery of phosphine gas. Phosphine is just pH 3. It's a gas that has been speculated to have been a biosignature on Earth. So microbes produce phosphine on Earth, and we detected, or it was said to be detected. It was allegedly detected on Venus recently using remote observations, although that study has been recently challenged. And so more about that if you want to Google it later, I guess. But we'll hopefully learn more when Da Vinci gets there. JUICE is another one of the missions that is going back to Jupiter. Jupiter Icy Moon Explorers. It's going to look at 3 of the 4, if not all 4 Jovian moons, so Io, Callisto, Ganymede, and the important one, the Europa, using mass spectrometers, a mass spec that's on there. Europa Lander is another mission that is specifically going to be looking for, again, those habitable environments and signatures of life by landing on Europa and looking at what could potentially be on the surface. There's this really cool idea that the surface of Europa is made out of these salts or stuff that could be coming from the subsurface oceans or even brine pockets that are in the ice shells. So we'll find out once we get to Europa. These are instrument concepts. Actually, this one is not a concept anymore. It's been selected to go on Europa Clipper, which is the companion orbiter that's going to be friends with Europa Lander. It's a multi-bounce time of flight mass spectrometer. It's capable of really cool isotope deconvolution to determine what we can actually see when we get to Europa. EMILI is another one of those really cool comprehensive instrument. By comprehensive, I mean it's got a lot of parts in it. So it's able to do liquid processing for the first time and ever, possibly? No, not ever, because we've done other types of liquid processing on other planets. But it's capable of separating liquid materials via capillary electrophoresis. So just by kinetics of your electrons of compounds that have been ionized. And it's also capable of gas chromatograph. So another one of our favorite separation techniques. And it's coupled to an ion trap, which is going to hopefully achieve heritage by the time ExoMars flies. And lastly, the future generation mass spectrometer, an ORBITRAP instrument. We've miniaturized it to enough that you can hold it in your hand. And it's one of the highest resolution mass spectrometers that have been built so far for space applications. That's going to allow us to really hone in on exactly what the masses of the molecules that you're seeing and the fragments, what their fragments are made out of. So that's all really cool and super exciting. And I will end my talk sort of here, going back home to where we are, I like to think of agnostic biosignatures as our future approach, or current and future approach to life detection. But we can't avoid this. Even if we want to be as unbiased as possible, we can't avoid the observations that we have to make of life on Earth, because this is the only example. And so we need to start somewhere, and that somewhere is here, but we also need to relax our assumptions about what some of those life forms can be, what imprints it can leave behind. And does anybody know what this image is? Pale blue dot. This is that. Excellent. So yeah, this is the image of our planet again. Just a tiny version of the previous picture, taken on the Voyager spacecraft. So Carl Sagan was the one who specifically requested for the spacecraft to be turned around, and this image became really famous from what he said about the fact that all of the life forms, beings, anything that you can ever think of or dream of, ever had thoughts, all lived through this tiny little pixel. And yeah, with that, I'd like to thank all of my super awesome collaborators in the Laboratory for Agnostic Biosignatures and my funding sources. So thank you.